Hello, today we're going to be talking about proteins and their monomers, amino acids. So what are proteins? Proteins are made of, as I said, amino acid monomers. They often have, they're three-dimensional. They have very unique and diverse shapes. In fact, they're so diverse that the body itself contains over 100,000 different types of proteins. Such diversity. Because of this diversity in structure, they have a lot of different functions. They can be enzymes, they can be structural, they can be used for signaling and hormones, a bunch of other things, in fact. All right, what are some examples? So all proteins are from the same 20 or so amino acids, but the proteins that they can make are much more diverse than that due to their combinations. And these are some examples of amino acids. So if we look back at the front slide, we can see what makes up amino acid. You have your amino group, the NH2, your carboxyl group, and your side chain. This side chain representing what's variable in each amino acid. So this is what changes. So for something so super simple like glycine, the only thing the only R group that exists there is an H. It's hard to see because of the way this molecule is written. But essentially, the only thing that's here in the H, the only thing in the R group is the H. This makes it super small, nonpolar, because there's no OH. Now serine, the hydroxyl group, a second one, in addition to the one it already has as part of its structure over here, and that makes it polar. Thirdly, aspartic acid. It has a carboxyl group. It's acidic, so it's negative charge. Also entirely dependent on the R group, as you can see here. Now dehydrogenase synthesis. So this is how singular amino acids bond together to create polypeptides. And this is sort of part of the process of protein synthesis that happens in the ribosome. It's sort of the first stage before we do folding and stuff like that. There's some interesting terminology, N-terminus, C-terminus. This just means where it goes along. So you can see the N-terminus being this amino group, so one of the H's from the N2H, or NH2, and C-terminus, carboxyl, so it's the OH right here. And that makes sense. This is how it is for all. And forms peptide bond, which makes sense, polypeptide. All right, so this is what happens after we do some dehydrogenase synthesis, and that is the sort of structural formation. Sort of four sequences: primary, secondary, tertiary, quaternary. And it sounds more complex than it is. So primary is just the linear structure amino acids, kind of like what we saw here. Secondary, you do some local folding, some interesting names for this: alpha helixes, beta sheets. Tertiary, it starts to become three-dimensional, sort of the things fold upon themselves. And then quaternary takes all of the tertiary things and bonds them together. So, yeah. All right, globular proteins. So globular means like globe-shaped, and this is important for sort of what it does in the body. So hemoglobin, for example, it's found in blood, and it's responsible for transporting oxygen. And it is globular in shape because of its environment. So the outside is hydrophilic, so it's polar, which makes sense because you want to be able to move in blood. But the inside, so what's carrying is nonpolar, hydrophobic. And this sort of makes sense. It's kind of like the kind of semi-permeable membranes in cells we see before in phospholipids. You had sort of different properties being used on different sides. All right, nitrogen. So sort of the key thing key element needed for these amino acids is nitrogen. Of course, we have the other cycles. We have carbon and phosphorus, and they're also useful. I mean, carbon's in everything, but nitrogen cycle is particularly important for amino acids and then nucleic acids. As we saw with the previous cycles, lots of different processes, organic and inorganic. There's a lot of stuff happening. All right, importance in life. So proteins, as I said before, very diverse, over 100,000 of them. And because of that, they're really valuable because they can accomplish things that not many other things can. Their sort of unique shape aspects can allow them to be used in different circumstances. And this is also defined by the R group. So you have even more complexity. You have the complexity of amino acids, then the folding, and then, yeah. All right. Thank you for listening.